This study was focused on the identification of beneficial bacteria associated with submerged plants. It has been shown that the microbiome of a plant can contribute to its survival rate. I wanted to see if I could find any microbes that were associated with a plant that was living in a submerged environment, and if those microbes had a direct effect on whether this plant was more able to survive in this environment than one that, say, did not have those microbes. So specifically, I was looking for at the roots of plants, as they had been shown to not grow as well while submerged, along with plant growth promoting rhizobacteria and endophytes, which have also been shown to increase the survival of plants living in submerged environments, as they can help with nitrogen fixation, ACC deaminase production, and pathogen interference. So in order to find the different species of bacteria that were associated with each of the rhizospheres and these rhizospheres of these plants, I went to Eastlake Park and got 3 different samples, which are labeled right here in this figure, SL3, SL04, and SL05, and then went through sample processing, culturing on both 1% LB and 100% LB plates, separation of the individual bacterial colonies, from which we were able to isolate the DNA that could be sent off for sequencing, along with PCR amplification. So from these different experimental procedures, I was able to collect information on the different bacteria that were isolated, and these are some of the pictures of the individual colonies. For the most part, these correspond to the different bacteria species down here, the most common match based on the percent ID from NCBI. And then I also listed the important characteristics that each of these bacterial species had on plant growth. The most important growth promoting factor that I found throughout all of these different species was that 7 of the 11 unique bacterial species in some way aided with salt tolerance, the most common one being through ACC deaminase production, which decreases the amount of ethylene in the plant and then therefore allows for salt tolerance to be more prevalent. Additionally, there were two different bacterial species that I found from other studies that have been shown to increase root growth. And I found that to be really interesting, as plants living in submerged environments don't generally have as good of root growth. So as there were a very high majority of the microbes that I isolated that had salt tolerance, I think it would be a good idea to further study if there's a higher proportion of these microbes in plants when they're living in a more salt-filled environment, or if I just had see if there's any correlation between the two, or if I could go back and get a sample from the lake that I took these from and see if there's a high salt concentration found there. So overall, it was definitely successful in the sense that I was able to identify quite a few different bacterial species that were associated with submerged plants and promoting their growth.